This case report describes the first documented instance of a Piscichuvirus-associated central nervous system disease in a free-ranging American alligator. It also represents only the second report to identify piscichuviral disease in any free-ranging reptile. The American alligator, Alligator mississippiensis, plays a crucial role in the southeastern United States, holding ecological, agricultural, and cultural significance. However, our understanding of diseases affecting these animals, particularly in the wild, is limited. While inflammation of the central nervous system is not commonly reported in crocodilians, there have been cases of meningoencephalitis linked to infections like West Nile virus and crocodilian herpesvirus in farmed alligators and crocodiles. Information about infectious diseases in free-ranging crocodilians is scarce. Shuvirids, a family of negative-sense, single-stranded RNA viruses, have recently emerged as potential pathogens in vertebrates. Initially discovered in a 2015 study focusing on arthropod viruses, shuvirids have primarily been identified through metagenomic studies of invertebrates, limiting our understanding of their disease associations. Recent research, however, has linked novel shuvirids within the genus Piscichuvirus to lymphoplasmacytic meningoencephalitis in free-ranging aquatic turtles. Microscopic, genetic, and in situ hybridization analyses have confirmed the presence of viral nucleic acids in central nervous system lesions in these turtles, providing the first concrete evidence of shuvirids causing clinical disease in vertebrates. In December 2022, a free-ranging subadult male American alligator in Clay County, Florida, displayed unusual behavior, remaining in a public location for approximately 2 days without moving. Recognizing the abnormality, an experienced trapper brought the alligator for clinical examination. The examination revealed severe stupor, tetraparesis, weakness in all 4 limbs, and an absence of proprioception, the sense of body position. Based on these findings, a lesion localized to either the brainstem or diffusely within the brain was suspected. Due to the severity of the neurological disease and a poor prognosis, the alligator was euthanized and a necropsy was immediately performed. While the alligator exhibited good muscling, it had a significantly atrophied coalomic fat body, which is the fat deposit in the abdominal cavity. No other significant gross lesions were observed. Samples from all visceral organs, the brain, and segments of the spinal cord were collected for further analysis. Microscopic examination revealed significant lesions confined to the central nervous system, affecting all regions of the brain and spinal cord. These lesions predominantly impacted the gray matter, but also extended into the white matter. The observed changes were interpreted as severe lymphohistiocytic meningoencephalomyelitis, indicating inflammation of the brain, spinal cord, and their protective membranes. To investigate a potential viral cause, next-generation sequencing was conducted on a frozen brain sample. The sequencing data, after removing the alligator host sequences, underwent de novo assembly and analysis. This analysis revealed a 10,814 base pair genome predicted to encode 4 open reading frames. Comparison with existing viral sequences showed the highest nucleotide identity, 92.3% to 97.2%, to a piscichuvirus found in a free-ranging alligator snapping turtle. Further phylogenetic analysis, based on the large protein gene sequences from 48 members of the Shuviridae family, confirmed the alligator virus as a sister virus to the alligator snapping turtle freshwater turtle neural virus 1 isolate. This finding strongly suggests a close evolutionary relationship between these 2 viruses. In situ hybridization, a technique used to detect specific nucleic acid sequences in tissue samples, was performed to confirm the presence of piscichuviral RNA in the alligator's brain and spinal cord lesions. This technique, using probes designed against the large protein gene of freshwater turtle neural virus 1, revealed a widespread, strong signal for piscichuviral RNA in neurons, glial cells, and occasionally ependymal cells of the alligator's brain and spinal cord. These findings further strengthened the link between the identified piscichuvirus and the observed central nervous system lesions. The clinical signs and microscopic lesions observed in the alligator closely resembled those reported in marine and freshwater turtles previously diagnosed with piscichuvirus infections. This suggests that piscichuviruses may cause similar neuropathology in different reptile species. It is important to note that while the central nervous system lesions in the alligator were indicative of a viral infection, similar inflammatory patterns have been observed in alligators and crocodiles with West Nile virus and crocodilian herpesvirus 2 infections. Therefore, a definitive diagnosis of piscichuvirus infection relies on molecular diagnostic methods. Figure 1 provides a visual representation of the meningoencephalomyelitis associated with the piscichuvirus infection in the alligator. Panels A, B, and C showcase the presence of prominent perivascular cuffs composed primarily of lymphocytes and histiocytes, a type of immune cell, surrounding blood vessels in various brain regions. These cuffs are accompanied by widespread gliosis, an increase in glial cells, which are non-neuronal cells in the nervous system, and scattered inflammation throughout the brain tissue. Panel D demonstrates the strong, intracytoplasmic, within the cytoplasm of cells, hybridization signal for piscichuviral RNA in neurons and glial cells, confirming the presence of the virus in these cells. Panels E and F illustrate similar inflammatory changes and hybridization signals in the spinal cord, further indicating widespread viral involvement in the central nervous system. Table 1 provides a detailed annotation of the piscichuvirus genome detected in the alligator's brain, including the positions and predicted functions of the 4 open reading frames identified. It also shows the high nucleotide identity between these open reading frames and those of the freshwater turtle neural virus 1 isolate from the alligator snapping turtle, further supporting the close relationship between these viruses. Figure 2 is a cladogram that depicts the evolutionary relationship between the alligator piscichuvirus and 48 other members of the Chiviridae family based on their large protein gene sequences. The alligator virus is clearly grouped with the freshwater turtle neural virus 1 isolate, confirming their close relationship within the genus Piscichuvirus. This figure also highlights the genetic diversity within the Chiviridae family and the distinct clustering of different viral species. The discovery of highly similar viruses in an American alligator and an alligator snapping turtle, species that share similar freshwater habitats, suggests that these viruses might represent a single viral species. This observation challenges the previous classification that grouped marine and freshwater turtle viruses as a single species, primarily based on genetic similarity. The fact that the alligator and the alligator snapping turtle, both infected with similar viruses, were found in relatively close proximity but 13 years apart raises intriguing questions about the persistence and transmission dynamics of this virus within the ecosystem. Further research is needed to understand the potential impact of this virus on reptile populations in these wetlands. The ability of this piscichuvirus to infect both crocodilians and turtles highlights its broad host range. Few viruses are known to cross such taxonomic boundaries, particularly in aquatic environments. One example is frog virus 3, ranavirus rana 1, which can infect select fish, reptile, and amphibian species. Given that turtles are part of the alligator's diet, transmission through ingestion is a likely possibility. This case report significantly contributes to our understanding of piscichuviruses as potential pathogens in reptiles. It emphasizes the need to consider these viruses as a differential diagnosis for neurological disease in both wild and captive turtles and crocodilians. Future research is crucial to unravel the transmission pathways, host range, and potential impact of these viruses on reptile populations. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.